Good morning and good afternoon, everyone. For those of you on the East Coast, welcome to another SoCalGas RD&D research webinar, and thanks for taking time out of your busy schedule to tune in today. My name is Tyler Nguyen, and I'm a project manager at SoCalGas. Our team, Gas Operations RD&D, operates within the Gas Research and Technology Division, which is a key component of the broader gas engineering organization. This structure enables us to align our innovation efforts with strategic engineering priorities across the enterprise. Today's webinar is being recorded. I'm coming to you live from sunny Southern California. Today, we're excited to welcome Mark Teagle and Dr. Yuri Van Haren from BioVind's, who will be sharing their expertise and insights with us. Now, I'd like to take a moment to clarify the title of today's webinar. The focus of this project is specifically on detecting microbes that could contribute to corrosion. SoCalGas would also like to express our gratitude to the California Energy Commission for their support and funding of this project. In today's webinar, you will have an opportunity to learn about the future of microbiologically influenced corrosion detection and management. The research team will compare commonly used microbial test approaches, including culture-based techniques, ATP testing, and next-generation sequencing. The team will also introduce a novel DNA-based platform designed for on-site microbial identification within 60 minutes. This emerging approach has the potential to enable faster, more reliable insights into microbial activity, opening pathways for improved corrosion rate prediction, treatment evaluation, and targeted mitigation strategies. At the end of the presentation, we hope that you will gain an understanding of how advanced molecular microbial detection methods that could support more effective and resilient corrosion management in the future. I'm going to come off camera just to save on the bandwidth. Before we get started, I'd like to quickly walk everyone through the GoToWebinar toolbar, which should be visible on your screen. This will help you navigate the features and make most of today's session. On your screen, you should see a small control. First, take a look at the small toolbar labeled Handout. Click the small arrow next to the word Handout to expand the window. There you should see a copy, which we are happy and excited to share with you, our 2024 SoCalGas RD&D Annual Report. We're currently working on the 2025 Annual Report, and we'll share that with you at a later date on our website. The next toolbar you'll see is labeled Questions. Again, click the small arrow next to the word. Here you can answer any questions you have during the presentations or review them during the Q&A sessions after the presentation. After the presentation and during the Q&A, we'll also open up the discussion for verbal questions. Click the raise icon to be called on during the Q&A session. And when you are called on to ask a question, we'll first grant you access to unmute. Then you should manually unmute yourself. Please go back on mute after asking your question. You may also have to change or update your computer settings to enable your microphone. So now is a good time to take a look and check those settings if you like. I want to share with you our commitment to the community. At SoCalGas, our commitment to customers goes beyond delivering natural gas. We strive to provide safe, affordable, and reliable service while actively supporting the communities we serve through innovation, sustainability, and social responsibility initiatives. Our RD&D team actively engages with community-based organizations across our service territory and throughout California to foster collaboration and innovation. We proactively reach out to researchers, entrepreneurs, and businesses in environmental and social justice communities to ensure inclusivity and broad participation in our programs. In addition to sharing information about our research funding opportunities, whether through SoCalGas or our research partners, we also publish results from our RD&D projects to inform and educate the public. Furthermore, we also track and report our equity engagement progress in the annual report to ensure transparency and accountability. The values of SoCalGas RD&D program aligns with the company's overall mission, which is safe, reliable, and affordable energy delivery today, ready for tomorrow. The SoCalGas RD&D program has three core pillars to help support that mission: science, synergy, and equity. Our team of experts in science and engineering work with the top researchers around the country and world, and other utilities and funding agencies to help develop new technologies that support the affordable access to clean, safe, and reliable energy for all Californians. To give you a quick overview of our research program structure, the SoCalGas RD&D program is currently organized into two key areas that span the entire supply chain. Low carbon resources team focuses on renewable gas production, including renewable natural gas and hydrogen, plus initiatives concentrating on the distributed power generation and microgrid technology. Additionally, our low carbon resources team covers thermal solutions from residential to commercial and industrial scale. Gas operations supports transmission, distribution, and storage operations through innovations that enhance pipeline, public and employee safety, while maintaining system reliability, increase operational efficiency, and minimize greenhouse gas impacts to the environment. The program also facilitates technology development driven by emerging regulatory requirements. The primary goal of gas operations are to develop, test, and introduce new gas technologies, operation technologies that are beneficial to ratepayers, public safety, and the environment. You can click on the QR code to connect with our programs to find out more about specific projects and results. So the best way, aside from reaching out to us directly, if you have any very specific questions, is to check out our annual report. In 2024, the SoCalGas team manages 226 active projects, which is a decrease from the previous years. On average, the SoCalGas RD&D program has a leverage of $7.10 in 2024. What that means is that for every dollar that SoCalGas invested in RD&D projects, $7.10 came from our external sources, which are partners, grants, and consortiums. Within the gas operations, we're getting a leverage ratio of $13.50 in 2024. What that means is that for every dollar that SoCalGas invested in projects under gas operations, $13.50 came from our external partners. In 2024, the team completed 70 projects and initiated 42 projects. This level of co-funding allows SoCalGas to continue to participate in valuable research at an affordable cost, which ultimately benefits our California ratepayers. If you would like to learn more about the RD&D program, please take a look at our 2024 annual report, including in the handouts or on our website. To better connect with the diverse communities we serve, we've also produced a Spanish version of the report. So both English and Spanish copies are available on our website in case you want to check them out in the future. And speaking of our website, you can access it by scanning the QR code on the screen. In addition to the annual report that I mentioned, you also can find information about our upcoming webinars and news about our R&D program. If you have any questions or research suggestions, please reach out to us via email at rddinfo@socalgas.com. Another good way to keep up with the SoCalGas R&D team is to follow us on our LinkedIn page. This QR code will take you to that page, and that's where you can see our regularly published posts about our research and outreach activities. We're also on Empower Innovations, which is an online networking platform initiated by the California Energy Commission. We invite you to sign up for our R&D newsletter, published quarterly. You can also easily subscribe using the QR code shown here. So now, it is my pleasure to introduce today's guest speakers, Mark Tico and Dr. Yuri Van Haren from BioVind's Incorporated. Both bring exceptional expertise and insight to our discussion, and we are honored to have them share their perspective with us. Mark Tico is the Chief Financial Officer of BioVind's Incorporated. He has over 20 years of experience in finance, compliance, and education, including senior leadership roles in the banking industry and extensive work with startups in biotechnology, intellectual property, and advanced hardware development. At BioVind, Mark leads the financial strategy, operations, and investor engagement as the company develops a portable microbial detection platform. Dr. Yuri Van Haren will be presenting the research study today. Dr. Van Haren has a PhD in molecular biology from the University of Maryland. He has over 10 years of experience working as a molecular biologist. His experience includes a broad variety of molecular genetic techniques, including the isolation and purification of genetic materials, DNA amplification, cloning, and the design of genetic amplifications and cloning assays, as well as substantial hands-on experience working with microbes in a human health and environment setting. At BioVind, which Dr. Van Haren is also a co-founder of, he leads the research development efforts exploring a novel method of on-site microbe detection. So welcome, Mark and Yuri. At this point, I'd like to turn it over to Dr. Van Haren, who will lead us through this presentation.
Yes, thank you, Tyler. Can everybody hear me okay? If not, please let me know. But yes, I appreciate that wonderful introduction, and we appreciate the opportunity to talk to everybody today about our new product for detecting microbes that might cause corrosion in oil and gas. So today, we'll start talking a little bit about our company, who we are, and what we do. I'll give a little bit of background on MIC, what it is and where you might encounter it. We'll talk a little bit about some of the currently available testing methods that people use to detect microbes that cause corrosion and why there's a need for alternatives. And then finally, we'll go over our approach to microbe detection and why we think it can, be an improvement to what's currently available. So who are we? So we are BioVind, right? Tyler already gave some introduction to me and Mark, but I also wanted to highlight our CEO, Joe, as well as one of our co-founders, Abehi, and then our full-time research scientist, Dr. Chris Lavello, who's working in our lab in San Diego to make sure that what we're working on actually works. We also wanted to highlight Tom Pickthall. He is the president of Enhancco, and he is essentially our industry expert, right? So he has been in the industry for many years. He has deep understanding and deep knowledge of what happens in the industry, what goes on, what people currently use to test for microbes. So he is a great advisor and a great asset for our team. And then, as Tyler mentioned, this project is funded by the CEC, or the California Energy Commission. So we want to thank them for providing the funding for us to do this work and to build this device that can help people detect microbes in oil and gas. I also want to thank all the folks that are on the technical advisory committee for the CEC. these folks are industry experts that provide us with the feedback to make sure that we stay on track and make sure that what we're doing actually is going to be beneficial for people. So with that, let's get into MIC. So what is MIC? So MIC is microbiologically influenced corrosion. It is corrosion that is initiated or influenced by microbiological activity. So this can generally occur in one or two ways. Either direct corrosion, where microbes use available electrons at the metal surface as an energy source to cause corrosion, or you get indirect corrosion, where corrosion occurs due to the byproducts that are generated as part of the microbial growth. So when and where might you see MIC, right? This corrosion that is induced by these microbes. So this kind of happens pretty much at all kinds of materials, right? So it can happen with alloys, including aluminum, carbon steel, stainless steel, brass. It also happens with non-metal materials such as concrete and plastic, right? Cause degradation there. But right now we're mostly focused on corrosion with these metals. A really important part of MIC is moisture. So microbes essentially need moisture in order to survive and exist, right? So if you don't have moisture in your system, you're probably not going to need to worry about MIC. However, if there's moisture in your system, even just a little bit, microbes can grow there and they can start causing issues. Other things that are important are flow rate and operating conditions of your pipelines, right? MIC is generally observed at the bottoms of your pipelines, specifically in areas with low flow rate, disruptions or stagnant conditions. Dead legs are a good example of that, right? And basically, when there are these low flow rate conditions, microbes can more easily settle on the surface of your pipelines and start causing corrosion. That also brings us to our next point, which is biofilms, right? Biofilms play an important role in causing corrosion at the metal surface. So biofilms are these extracellular matrices that are created by microbes that basically protect them from the environment and create this kind of microenvironment to allow microbes to thrive and grow. So when you have these biofilms that are formed at the surface of your material, that area is going to be especially subject to corrosion. And another thing I wanted to highlight here was the complexity of corrosion, right? So corrosion is not as simple as you have microbes and therefore you have corrosion. There's many, many different microbes that are there in our environment. A lot of them can cause corrosion, but there's also a lot of them that don't cause corrosion, and there's even some that can inhibit corrosion, right? And there's generally kind of a complex interplay of different microbial species that live together in a community that actually cause the corrosion, right? So it's much more complex than just one microbe is causing corrosion. But there are specific microbes that are known to be more involved in corrosion. So I want to go over kind of the main culprits when it comes to MIC. And what are the microbes that generally tend to cause corrosion, and that when you're thinking about MIC, these are the microbes that you really want to be concerned with. So I have some examples here. There's many more, but I'm going to go over kind of the 5 main ones that you want to think about. So first, you have your SRB, or sulfate-reducing bacteria. These are microbes that use sulfate instead of oxygen for their respiration, and basically cause corrosion right at the metal surface. So this would be an example of that direct corrosion. We also have APBs, or acid-producing bacteria. These, as the name suggests, are microbes that produce a lot of acids as part of their metabolism, right? And the release of all these acids into the environment will obviously cause some corrosion, right? So this is an example of that indirect corrosion. You have IRB and MRB, which are iron and manganese reducing bacteria. These are microbes that, similar to SRB, cause corrosion right at the metal surface. You get your methanogens. So methanogens are microbes that produce methane as part of their metabolism and thus cause corrosion. And then finally, you got your biofilm forming microbes. So this is a quite important group, right? We talked about biofilm already a little bit. But basically, these microbes are microbes that create that biofilm that allows these microbes to settle on that metal surface and really start causing problems. But the main point here is that the interaction between these different microbes really does matter, right? It's usually not as simple as you have one group of microbes in your system only. Usually there's multiple different groups of microbes at a specific site that are interacting with each other to cause corrosion. So that was a really brief background of MIC and what corrosion is. So now let's talk a little bit about kind of what people are using currently to detect these microbes that are causing problems. and why there's a need for alternative methods that can tell you a little bit more about what is actually going on in your system. So when you're thinking about monitoring for microbes, right, and testing for these microbes, there's a few questions that you want to answer. So first, you want to know where these microbes are actually located, right? So you want to figure out where in your system do you have microbial growth. Is it upstream, downstream? Is it all kind of located in one area? Or is it spread throughout? So you want to figure out where microbes are growing. Next thing you want to ask yourself is what microbes are present. Are there SRBs? Are there APBs? Are there microbes that are forming biofilms, right? Which types of microbes are actually in your system? Are they maybe microbes that do not cause corrosion, right? So you really want to know what type of microbes they are. Another thing that you want to be concerned about is how many microbes are there. Microbes are pretty much everywhere, so it's not unusual to find microbes, but you really want to get an understanding of whether or not the amount of microbes that are in your system is high enough that it's going to cause issues and might start causing corrosion, or if there's just a few microbes and it's not really anything of concern right now, it's just something you want to monitor but not necessarily do any treatment for. And then lastly, you want to figure out what they are doing, right? And it kind of plays into all of these points, right? You want to figure out where are these microbes and are they causing corrosion, right? Are they forming biofilms? Are they producing acids, right? So you want to figure out what microbes are there so that you can get an understanding of what these microbes are doing in your system. And when you're thinking about monitoring for MIC, right, really the idea is that the more information you can get about the microbes that are there, the better, right? Because the more information you have, the better of an understanding you can get what is actually going on in your system. You can do modeling and monitoring of your system over time under different conditions and really start getting an understanding of when your system might be at high risk of corrosion caused by microbes. So let's get into the testing methods for detecting these microbes. So there's three main methods that are currently being used in the field that I want to go over. So that includes ATP testing, culture-based testing, and molecular methods. So I'll go over all of these in detail. So starting with ATP testing. So ATP testing tests for the presence of adenosine triphosphate, ATP for short. So ATP is an energy molecule that exists in all living cells, right? So you have these tests that are these simple snap tests that you can take on site, and they give you results within minutes and tell you essentially how much ATP is in your sample. So you take a sample, you test it really quickly, and it tells you there's this much ATP in your sample, and that can then be used as a proxy to tell you how much microbes are in your sample. So it's a super easy test, and it tells you kind of roughly how many microbes are in your samples. But it does have some significant limitations in that it doesn't tell you anything about the types of microbes that are in your sample, right? So because it doesn't give you any information about the type of microbe, it can't distinguish harmful from non-harmful microbes. So you really don't learn anything about what's going on in your system. If you then want to get a little bit more information about what is actually happening in your system, you can use these culture-based testing methods. These are often referred to as bug bottles. And the way this works is you basically take your sample and you add your sample to these glass vials that contain this bacterial growth culture medium. Now there's various different types of bacterial growth culture medium that allow for the growth of different types of microbial groups. For example, you have media that allows for the growth of SRBs, you have media that allows for the growth of APBs, and therefore you can kind of get a rough understanding of what types of microbes are in your system. So the way this works is when you add your sample to these media bottles, right, and there's microbes in your sample, these microbes starts growing, and the growth of the microbes causes a change in the appearance of the media. So change either from red to yellow or from clear to kind of cloudy or black. And that tells you that there's microbes in your sample that are growing in this culture media. Another nice things about these culture media tests is that you can do serial dilutions, where you basically dilute your samples multiple different times and add them to these bug bottles, and that gives you a rough idea of how many microbes are there in your sample. So you can get a rough sense of, are there a lot of microbes or are there just a few microbes? And this is also the most commonly used testing method in the field. So this is something people are familiar with, and they're comfortable with this testing method, but it does have significant limitations. So this media can tell you roughly the type of microbes that's in your species or in your sample. So it tells you whether or not there's SRB or APB, but it doesn't tell you anything more. So it doesn't tell you the specific species of acid-producing bacteria or sulfate-producing bacteria that are in your sample. In addition to that, not all microbes actually grow in these culture media conditions. Methanogens is a good example of that. Methanogens don't grow in culture media conditions. So there's no way to test for the presence of methanogens using these bug bottles. So when you're using these bottles, you're going to miss a whole lot of microbes because not all microbes will actually grow in these bug bottle conditions. In addition to that, these bug bottles suffer from culture bias. Basically, what this means is that the stuff that's going to grow in your system, right, in the samples that you draw from your pipelines, your storage tank, or wherever, right, it's always going to be very different than the stuff that's going to grow in these culture bottles, because the environment in these two locations is so different, right? So you might have bacteria that grow really well in your pipeline or storage tank, right, and they're really happy there, and they're causing issues. But then when you take them out and put them in these bug bottles, they don't grow because they just don't like that environment, right? So if you do testing with these bug bottles, you're going to miss those and you're going to undertreat. On the flip side of that, you have specific microbes that don't really like the environment of your pipelines or storage tanks. So they might be there, but they might not be causing any issues. But then when you take these samples and put them in these bug bottle culture media, they love that environment. They start growing and growing, and you get this aggressive growth. And you're thinking, okay, there's a lot of microbes in my system, even though the microbes are not actually causing problems when they're in your system, right? So you can get these false positive and false negative results with these tests. And then another big downside of these tests is that obviously these microbes need to grow, and microbes don't grow especially fast, right? So it can take up to 2 to 4 weeks for these microbes to grow and start showing results. So the results you get back from these are not immediate. You usually have to wait several weeks to get your results back. So because of these limitations of these two tests, right, it doesn't really tell you much about the type of microbes that are in your system. People are oftentimes advocating to do more detailed tests that can tell you more detail about what microbes are actually in your system. And one of the methods they often suggest in the literature is these molecular testing methods. So that includes qPCR and next-gen sequencing testing. So what are those, right? So qPCR, you may be familiar with from COVID, right? These are tests that are often used to test for human disease. And they're super powerful tests, right? They can tell you exactly the type of microbe that is or isn't in your sample. The way these work is that they basically detect the presence of a species by looking at whether or not their DNA is present. So the way you want to do this when you're testing for microbes in oil and gas is you take a sample, you then have to send the sample to a third-party testing lab because they have to have the proper equipment and the trained personnel to be able to do these testing methods. So you send your sample to this third-party testing lab. Over there, they isolate the DNA and then use those assays to detect the presence of specific species of microbes. So you can test for example, specific species of SRB, and qPCR can tell you if it's there and roughly how much of it is there. So it's super sensitive, it's highly trusted, but again, it has limitations, right? So one of the big limitations of qPCR testing is that it cannot be done in-house, right? Obviously, these testing methods require complex equipment and trained personnel, so it's difficult to do them yourself. You always kind of gonna have to send them away to a third-party testing lab. Because you have to send them over to another lab, right? You kind of have to ship those samples, and then they get processed at that lab. And during this whole shipping and processing period, your sample might start degrading, right? So you might lose some of the signal that might otherwise be there in your sample. And another downside is that it generally takes a while to get your results back, right? So it can take weeks to get your results back from qPCR testing. If you have a really good lab that happens to be close by, you can sometimes get your results back in several days, but usually it takes several weeks to get your results back. And then the main problem with this testing is that it's just expensive and difficult to scale up, right? So even though it tells you a lot of information about what is actually in your sample, it's just so expensive that it's not really feasible to scale this up to be able to do regular testing across your system at different dates, right? So it's just not really feasible to use this as a monitoring system. It's more of kind of like a once in a while thing. In addition to qPCR, you also have next-gen sequencing testing. This is similar in a lot of ways to qPCR, but instead of having a single test, they can test for the presence of a single species or group of species. Next-gen sequencing takes a single test and basically can identify all the microbial species that are present in that sample. So in essence, it's the same as the qPCR test, right? You take your sample, you send it to a lab, they isolate the DNA, and they then do the sequencing testing in this specialized equipment and give you back a result that tells you exactly what's in your sample. So this is really the gold standard of detecting microbes in any sample. But it has similar limitations, right? It can't be done in-house. Microbial DNA might start degrading during the shipping and processing steps. And it can take weeks to get your results back, right? This generally takes even longer than qPCR testing. So weeks to months is not unusual when you're doing sequencing. In addition to that, it doesn't provide you any quantitative information about the microbes that are there. So it tells you the relative abundance of different species of microbes that are there within a single sample, but it doesn't tell you the abundance of microbes comparing two different samples, right? So if you're sampling site A and site B, next-gen sequencing can tell you which of those sites have more microbes, basically. It just tells you what microbes are actually there. And then finally, this is expensive and difficult to scale up, right? Similar to qPCR, it's not something that you can really do regularly because it just becomes prohibitively expensive. So those are kind of the testing methods that people are using in the field today, right? And when you're thinking about testing for these microbial species that might cause corrosion, right, there's a few things that you generally want to consider, right? You want to have a test that tells you enough information about these microbes so that you can get an understanding of what's actually happening in your system, right? So you want them to be specific. In addition to that, you want them to be simple, fast, and portable, right? So you want a test that you can take on site, do on site, and get results right away instead of having to send them out and wait several weeks to get your results back. And then finally, you want something that's cost effective. All right, so you want something that's not gonna break the bank and that you can kind of do regularly, right? You want to test that you can test multiple different sites on multiple different days relatively easily without, you know, investing a crazy amount of money. So when you're thinking about the currently available testing methods, right, when you think about these bug bottles, they give you some information about what's there in your sample. They are fairly cheap, but they're not simple, fast, and portable, right? So they're quite complicated and take a long time to get your results back. ATP testing is super cheap, super simple, fast, and portable, super easy to do, but it doesn't provide any information about what's there in your sample. And then on the flip side of that, you have your NGS and your qPCR testing, which tells you everything that's there in your sample, right? It gives you a lot of information, but it's quite expensive and it's not really simple or fast. So ideally, you want something that's right there in that middle, right? That sweet spot where it's, you know, provides you with information about what's there, it's cost effective and simple, fast, and portable. So that's where we come in, right? So this is our BioVind's solution to this problem of detecting microbes in your oil and gas system. So our aim is to develop a test kit for detecting specific microbial species or groups that is associated with MIC in oil and gas pipelines and storage facilities that can produce results within an hour. The test kit would test for multiple different targets in a single run and can be used by field techs without any specialized training. So what we're looking to develop here is a test that can give you lab quality specificity and sensitivity, similar to qPCR. It's portable, so you can take it on site and do testing on site. It's simple, so it's something that you know, you don't need specialized training to do. It's fast, so get your result right away within an hour. You can multiplex, so you can test for multiple different species with a single sample. And then it's low cost, right? It's something that you can do regularly without investing a whole bunch of money. So this is what our development looked like for this process, right? So we started with SoCalGas to go on site, do some testing with them using the currently available methods and collect some samples to take back to our lab to help in the development of our assays. So we then, in our own lab, started developing assays to detect some of these microbial species that you may be concerned with when you're thinking about MIC. In addition to developing these assays, we also developed a user-friendly sample preparation protocol. Once that was all developed, we tested it in the lab to make sure that it worked using this DNA from these target species of microbes. When we validated that everything worked, we went on site with SoCalGas, did a pilot demonstration to show that the test works on site. We got some feedback and we're doing additional refinements to the system now to get ready to go to market. So taking a step back here and talking about the development of these assays, basically what we did when we started building these assays against these microbes was to first figure out which microbes are people actually concerned with when they're thinking about this microbially induced corrosion. Right, and we basically identified 4 main high priority targets, right? So that includes those sulfate reducing bacteria, these acid producing bacteria, the methanogens, as well as Pseudomonas. Pseudomonas is an example of one of those biofilm forming microbes. And we created this list based off conversations that we had with people that work in the industry, based off of literature research and based off of testing that we did on these field samples. So we're focused initially on generating assays against these high priority targets. So when we built our assay, what this looks like, it's really similar to qPCR, where it essentially uses the DNA to be able to detect the presence of specific species of microbes. So what our test looks like is it has these assay reagents that include a primer sequence that can bind the DNA of specific targets species, right? So if you're testing, for example, for a specific species of methanogen, we have a primer that can detect that specific species of methanogen's DNA. And then once you add a sample that has that species of methanogen in there, the primer will bind to its DNA, and then the reaction begins and it starts amplifying this DNA. So it starts creating more and more DNA over time. We then have a molecular probe in our assay that can detect DNA and fluoresces when it binds DNA. So the more DNA is created, the more fluorescence you'll see from this probe. So what this looks like when we develop these assays is that when we have one assay that tests for a specific MIC-related species, right? And we added our target species onto our assay, we found that you see this nice amplification curve, where if you look at the change in fluorescence here, you see that the fluorescence increases as more and more DNA is generated. And that basically indicates that, yes, this specific species of microbe is there in your sample. If we added a sample with a different species of microbes, so one that we didn't test for, right, it basically did not show any amplification, right? Suggesting that this test can specifically detect the species that you are testing for. Another thing that we wanted to look at was to see whether or not these assays can identify the relative abundance of the microbes that are in these samples. So what we did is we created several samples with varying amounts of microbial DNA in there from our target species. And we found that based on the amount of microbes or microbial DNA that are in these samples, the speed of amplification changes. So you can see here that the sample with the highest amount of target DNA in the sample showed amplification the soonest, right? So based on the time of amplification, you can get a rough estimate of the amount of microbes that are in your sample. So if you're comparing multiple different samples, you can kind of get a rough idea of which sample has the most microbes in there. So once we kind of validated that and we found that this works, right, we can detect these microbial species and tell you roughly how many microbes are in your sample. We then wanted to get these tests ready for use in the field. So what that looked like for us is we have this cartridge. You see the picture here at the bottom, and this is our test cartridge, right? And this cartridge contains 20 wells that all contain a specific assay. So we have 2 control assays, and then we have 18 different test assays. So with one cartridge, you can test for 18 different microbial species or microbial groups. So you can take one sample and test for 18 different things simultaneously. So to get these cartridges ready, what we did is we took these assays that we built and we lyophilized them, meaning that we essentially freeze-dried these reagents into this stable kind of lyophilized beads. And this basically helped with the stability of these assays, makes them shelf-stable for about 2 years at room temperature. So it eliminates the need for any cold storages and makes it really easy to work with these cartridges, right? Because you can just basically throw them in the back of your truck, drive out into the field and do your testing. You don't have to worry about keeping your samples cold. You don't have to worry about keeping your tests cold. It's just super easy to work with. So this is what our completed kind of test kit looked like, right? We have our assay cartridge with those 18 different tests in there. And then we have our amplification device. So the device basically takes the cartridge and then starts the amplification process and detects the fluorescence from the molecular probe, right? And then the device has a screen at the top that can tell you in real time whether or not specific species of microbes are detected. So once you put your cartridge with your sample in there, it automatically starts running, and then it tells you in real time if specific species are detected. So super easy to use. And this is what that looks like in practice, right? And hopefully you can kind of see this okay. I know these videos never play super well, but basically you take your cartridge or you take your sample in a syringe, you deposit it into the cartridge, and then you take the cartridge, put it into the device, you close the device, and then it runs for 30 minutes. And at the end of the 30 minutes, it'll show you a final screen that tells you exactly which microbes were detected in your sample. So when we took this device on site to do testing, we mainly focused on testing liquid samples, so samples that you would collect from drips, for example, right? One of the things with these liquid samples is that they tend to be fairly dilute, right? Meaning that the microbes are not very concentrated. So one thing we recommend with liquid samples is to first concentrate that. So the way we do that is we take one of these samples and we filter it through vacuum filtration to concentrate all the microbes that are in the sample onto one of these filter papers. We then cut out the filter paper, put it in a specialized developed buffer called BoreLab buffer. We then shake up the sample for 20 minutes to allow the microbes to release and to expose their DNA. We suck the sample up into a syringe, then use that syringe to put it into the cartridge and into the device, and you're ready to go. So it's a pretty straightforward process. This is what that looks like in real life. Oh, let me play one video at a time here. So we start by collecting the sample, and again, hopefully you guys can see this okay, but basically you take your sample. In this case, we're doing a control sample, so it's just water, but it's the same for any sample that you collect from a drip or whatever. And you basically vacuum filter it to concentrate the microbes onto that filter paper. The next step is to take that filter paper and cut it out, and then take that filter paper with the microbes on it and put it into a tube that contains this BoreLab buffer, right? So you take out that filter paper, put it into that BoreLab buffer, and you then shake it up for about 20 minutes to release the microbes and to expose their DNA. After that shaking step, you basically draw up the sample into a syringe, and then you use that syringe to deposit the sample into the cartridge, and then it immediately dissolves the beads. So the assay is then ready to go. So you take your cartridge, put it into the amplification device, and as soon as you close the lid, it automatically starts running, and then within 30 minutes, it tells you exactly what microbes are there. So super straightforward process, right? As you can see from these videos, it's a bit messy still, so we're still looking to kind of optimize the process and make it as easy to use as possible, but it works really well already. So when we went on site, right, with our test, we obviously wanted to see whether or not it actually works to detect microbial species in these field samples. So what we did is we created the cartridge with just one test in there. So this one has a test to detect the presence of the sulfur-Vibrio, which is a specific species of SRB. Obviously, in future iterations, we have additional tests in a single cartridge, but for now, we wanted to only test for the one species. So the first thing we did on site is we created positive and negative control samples. So we basically had samples that we knew had that species of microbe in there, and one sample that we knew we did not have the species of microbe in there. And we saw with the positive control that has the microbe in there, you get these nice amplification curves, and the screen shows a positive result, right? Suggesting it works exactly as expected. With the negative control, you don't see amplification curves, you see a negative result on the screen. So the control samples performed exactly as you would expect. So the next step was to test field samples. So we collected several field samples and we tested 5 of those field samples. Out of the 5 samples, 4 of them turned up negative, basically showing that there's no desulfur-Vibrio microbes in those samples. One of these samples turned up positive, showing that yes, desulfur-Vibrio is present in this sample. And you can see the curves here. They look a little bit more messy compared to the control, just because there's other materials from those field samples in there that interfere with the fluorescence a little bit to make it messy, but it works just as well. The device did show up positive. We then wanted to validate that these results are accurate, right? So we took these exact same samples and did next-gen sequencing testing on them, and that testing confirmed that the 4 samples that showed up negative for us also showed up negative with NGS. And then the sample that showed up positive for us confirmed that it indeed had desulfur-Vibrio in that sample, right? Basically showing that our test can accurately detect microbial species when it's there and not detect it when it's not there. So that's great, right? The test works well on site. The next thing we tested was, or not the next thing, but something we also tested while we were on site was usability of the BioVind test. So basically we measured the total hands-on time as well as the total time to results when using these tests. And we found that with the BioVind test, you have a total hands-on time of about 5 minutes and 30 seconds and a total time to results of about 75 minutes. When we compared this to some of the other methods that are out there, right, really we saw that the only test that can outperform our test is ATP testing, right? ATP testing is just so easy and so simple. It's super fast, but again, doesn't give you any information. But when you compare it to bug bottles, PCR, or sequencing, you see that we are much faster and provide results within an hour and a half, whereas the other ones take several days or weeks to get your result back. So performance is looking pretty good there. So when we kind of look at this as a whole, right, and look at the different testing methods that are available, ATP, culture testing, qPCR, sequencing, and then that new BioVind test that we're working on, and look at whether or not it can do these things that you want out of a test that can detect microbes, right? So can it detect the microbial abundance? Can it detect microbial diversity? So what microbes are in your sample? Is it low cost? Can it provide results to you quickly? Is it portable? Can you take it on site? Can it detect multiple different species within a single sample, right? And we found that BioVind is really the only test that can tell you about the microbes that are in your sample without sacrificing cost and speed of your results, right? So obviously, right, having a test that can tell you what's in your sample and provide you details about what's actually going on in your pipelines or in your storage tanks or wherever you're doing testing, right, and do that quickly, same day, easily, and at a low cost is super powerful. So some of the benefits that you have with a test like this are obviously it can help you do any modeling, right? So with AI taking over the world, everybody's moving in a direction of we want to use AI to do all this modeling and whatnot, right? And obviously when you're thinking about how do I model MICs and microbes growing in these complex systems, right? The first thing that you need is a whole bunch of data, right? And having a test that can create a whole bunch of valuable data about the microbes in your system and do it relatively cheaply allows you to generate the data that would allow you to do these modeling so that you can kind of track how microbial growth changes depending on operating conditions, depending on weather conditions, and kind of use that information to help you do predictive modeling to kind of get a sense of when your system might be at a higher risk for microbially induced corrosion. It also helps with treatment, right? So when you're thinking about creating these treatment plans for when you want to treat for microbial growth, right? Getting a better understanding of the types of microbes that are in your system allow you to be much more diligent when you're creating these treatment plans and allow you to be much more specific. So instead of treating really broadly for just a general chemical that kills all microbe that's really harsh and really expensive, right? You can be a little bit more targeted with your treatment. It also kind of will help you understand what is going on as a whole when it comes to these microbes, right? So like I mentioned, there's many different types of microbes. And, really, it's important to understand the different types of microbes and what they're doing, right? So there's microbes that cause corrosion, that don't cause corrosion. There's microbes that can inhibit corrosion. So being able to do this more detailed testing can give you a lot more information to really understand what's happening. It can help with mitigation. So if you are doing any mitigation or treatment, these kind of testing can help you determine the effectiveness of these treatments, right? Where you can basically do testing before and after to see whether or not your treatment was effective, to see whether or not it's killing only SRB, if it's killing everything, or only like one type of microbe, right? So it gives you a lot more information and can basically tell you if your treatment is effective. It allows you to be much more proactive. Basically, right, instead of waiting for your results to come back for several weeks and kind of chasing the problem or testing and waiting for your problem to get worse before you know if it's actually a problem, right? Getting the results right away allows you to be much more proactive in your treatment. It helps you save money. So biocides, these chemicals that treat for microbes, are quite expensive, as well as being quite environmentally harsh, right? So allowing you to understand when you need to treat, but more importantly, when you don't need to treat, really can help you save money on these biocides, as well as save the environment a little bit by using less of these dirty chemicals. And then finally, it can help with any failure analysis, right? So if you do have any failures, basically, having this test available to you that you can deploy at a moment's notice allows you just to go on site and do some quick testing to at least let you know whether or not microbes may have been involved in any failure that has occurred. So a lot of benefits to this testing. So that's why we're working on this, right? That's why we're developing this test. So we're looking pretty good to date when it comes to development. There's still a few different things that we are working on or working towards, but what we have achieved to date is that we've developed these assays that can detect specific contributing microbial species that contribute to MIC, right? So that includes species such as SRB, APB, methanogens, as well as some of these biofilm-forming microbes. In addition to that, we've developed the hardware. So we engineered our single-use cartridges and amplification device and optimized them for harsh field conditions, as well as long shelf lives. And then we've done that real-world testing, right, where we took everything on site, did the testing, and showed that we can detect microbes in field samples and tell you whether or not it's there or it's not there. But there's still work to be done, right? So there's some ongoing work where we are still looking to expand the assay library that we have, right? So right now we have tests for 6 different species of microbes. We obviously want to increase that number, right? So we want to develop additional tests for as many microbial species that are involved in corrosion as possible, so that so that people can test for pretty much any species that they would like using our testing method. We're also working to further refine our device and cartridges to basically optimize the design of it and make it as easy to use as possible. We're looking to do additional field trials and capture more data. Right, during our last field trial, we tested just one of our assays, so we want to do additional testing to basically be as rigorous as possible with our testing to really make sure that everything works as intended, as well as just collect more data on all the different tests that we are developing. Another thing that we're looking to do is to figure out how we can use our tests to integrate them with any biocide treatment plans that people generate, right? So how can our test really help people optimize their treatment, and kind of what data can we provide to really help with that? Then we're working on go-to-market strategy to get ready for a commercial launch, hopefully next year. And then we're working on scalability with our manufacturing partners to go from kind of a prototype level of development to go to large-scale manufacturing. And then the last thing here is that we're working on a data and analytics platform to basically allow you to upload your data that you get with our device onto this platform, and then use that data to kind of monitor your systems over time, or monitor different locations within your system to basically allow you to do that analytics in our software. So that's it. That's what we're working on, and that's why we're excited about this. So we'll have some time for Q&A now. So Tyler, you want to take over here again?
Sure. All right. Thanks, Jerry. A lot of good information. So we have time to do some questions in for the Q&A session. So again, if you have questions from the audience, just raise your hand. Use the raise hand icon to be called on. And once you're called on, unmute and then just ask questions. And then once you're done answering your questions, please go off and mute yourself again. So are there any questions? If not, we do have some questions that we received prior to the webinar. So. All right, looks like we don't have any questions from the audience, although we do have some questions. So the first questions that we received is from Occidental Petroleum Corporation. So the question is, what is the detectability limit and the field application requirement? So Yuri, do you want to answer that?
Yeah, absolutely. So the limit of detection, right, for these tests varies by test, but generally what we see is that the sensitivity of these tests is similar to qPCR or more sensitive than qPCR, right? So you can detect really, really small amounts of microbes. The the field requirement for testing's limit of detection, right, is not exactly known, but essentially you want to be able to detect microbes when they're there in sufficient amounts to cause problems, right? So when there's a few microbes floating around, but they're not really growing or dividing, you're not really going to worry about them. So you don't necessarily want to detect every individual microbe, which is obviously not something we can do anyways. But when there are a good amount of microbes in your sample, you are going to have to be worried about corrosion, and at that point, you want to be able to detect it, and that's usually what we see that we can do.
Okay, great. Thank you, Yuri. And the second question we have is with RSI pipeline solutions. And the question is, with regards to sample limitations, does it work on liquid samples, solids? What about liquids consisting mostly of oil with some entrained water?
Yeah, good question. So I think I talked about this a little bit already, but basically what we did for most of our pilot testing is work with liquid samples, right? A lot of these liquid samples did have oils and kind of contaminants in there. And what we found is that usually when you do the filtration, you basically pull out all the liquid to concentrate only the solids onto that filter paper, and that helps a lot to kind of get rid of a lot of these contaminants. In addition to that, we developed that specialized buffer that I talked about, that Bohr Lab buffer, to help resuspend the microbes, and that has some chemicals in there as well that kind of neutralize some of these harmful chemicals that could otherwise kind of interfere with the amplification. So we haven't seen huge issues there. to be fair, we haven't tested crude oil samples quite yet, so that's something that we'll have to look into in the future. But with the samples that we have collected, we do see that we don't have any issues with any interference. For solid samples, this process should be a little bit simpler. Again, we've been focused on liquid samples, so we don't have kind of tested the solid samples yet, but it is something that we're working towards. But basically for solid samples, the nice thing is that you don't need to go to that extra concentration step, because usually if you're doing, for example, a coupon pool, and you can basically just swap those coupons and go straight into the bore lab without having to do the filtration. So it should be fairly straightforward.
All right, so the next question is from Energia, I hope I'm saying it right, BB Incorporated, and the question is, have you explored the effects of hydrogen sulfide and oxygen in microbially influenced corrosion?
So we are mostly focused on the detection aspect, right? So we're basically working on detecting specific microbes. We're not necessarily doing research on how corrosion actually happens on site, right? And then the chemistry behind that. So we're mostly focused on microbial detection. So no, we have not looked at that directly, but I do know that those do play a role in causing corrosion, right, in your systems. Sulfate-producing bacteria is an example of that, right? They work or they create these hydrogen sulfides that can, cause that complex chemical reaction that causes corrosion on your metal surface.
All right, so we're running short on time, so I'll just take one more question. And this question is from Ohio Linux Company. So the question is with regards with issues with contamination, false positives, how can we test for and quantify all species involved in the mix?
Yeah, so with regards to contamination and cross-contamination, right, there's always risk for cross-contamination, right? But this is true for any testing that you do. The idea is that you just use general good practices to prevent cross-contamination. One of the benefits of our test over, for example, blood bottle testing or ATP testing is that it's species specific. So if you do contaminate your sample by like coughing in it or something, or you stick your finger in it and a whole bunch of microbes get in your sample, it's not going to get detected using our test, because our test only detects that one specific species that causes corrosion. So you don't really have to be quite as worried about contaminating your sample. With regards to testing for all the microbial species that are in your sample, if you want to do that, you really have to do qPCR testing. We can do 18 different species at the same time, and the idea is to basically get the 18 most relevant species, right? So the 18 species that are most likely to cause issues in your system, and test for those. But we can't test for all different microbes, because there will be hundreds of thousands of different microbes that are present in the system at any time. So it'll be really difficult to test for all of that with a portable test. You'd really have to use sequencing for that, and it's quite expensive. But one of the things that we are looking to do, right, is to basically work with customers and basically go on site, do testing with them, take samples, do sequencing testing to figure out what microbes are in their system, and then develop these customized cartridges for those specific systems, so that we can make sure that we're testing for the species that are there and that are causing issues.
All right, so for the remaining questions, we don't have time to address all of them, but what we'll do is that we'll review all the questions that were submitted.
We have a few more moments, and if you want to, if you
You
or
have more
Yuri
time?
wants to address
Okay,
anyone. Yeah.
so,
Yeah, let's do it.
all right, so let's go on with the next question. Okay, you look excited about it. Okay. All right, so the next question is from Angel Investor. So how big of an issue is MIC for oil and gas? How will rapid testing change the cost that you face, change the actions that you take, for instance, with biocide flushing?
Yeah, I'll pass this one on to Mark. Mark, you want to talk about this as the CFO here?
Yeah, actually, we had this question yesterday when we had Tom on the line with us, and his response was basically, it's everywhere. Anywhere where there is water, there is concern about microbes that are inducing that corrosion. So the main advantage here is that with our system, you can efficiently identify those microbes in the field within the 30 minutes and be able to identify or be able to develop your mitigation strategy right then and there and targeting those specific microbes. So you can also do kill studies, by the way. So if you've got specific samples and you pull those and you can test your different, biocides, quantity of biocides, anything like that, you can start tailoring it to just the needs of that specific area of your system.
All right, so the next one is actually
Tyler, just a quick, can we ask a question from the chat? We have a few questions here. I'm going to go ahead and ask the one. Can you quantify the microbes? Otherwise, answer the question of how much is there.
So not exactly. So we can do pure quantification in that sense where we can tell you exactly how much microbes there are, but we can do relative abundance. So basically what our system does is it can tell you based on the time of amplification, right, how soon the amplification start, roughly how many microbes are in your sample. So the idea of quantification with our system is that we would create kind of a standard curve and use that to give you an estimation of roughly how much microbes you think are in your sample. Another thing is to just basically compare two different samples or basically two different samples, compare them side by side to see, which one shows amplification earlier, and that one will have more microbes in there. But we can't do pure quantification, but we can give you a relative estimate of the amount of microbes that are in your sample, if that makes sense.
Yeah, and the idea here is this is a field deployment, right? You're out in the field. You don't want to have to worry about getting all those quantifications and then doing your calculations, things like that. You know that if it pings early and it says, hey, within 13 minutes you get SRBs in there, you got a problem. If it's down at 29.5 minutes, you can take it from there.
Thank you. Diane Saber, I see that you posted a question and you also mentioned you raised your hand. Sorry, we missed the raised hand. Do you want to unmute and ask the question, or would you like me to go ahead and ask it for you? We'll give her a few moments to see if she unmutes. Okay, I'll go ahead and ask it. She said the test is a modification of a test which is used in medical environments and can be useful. However, we are looking at bacteria in the gas rather than on the surface of the pipe. There is, as far as we know, a sufficient indicator for corrosion. There is no data to indicate the presence of bacteria with corrosion. Does this account for dead cells versus live cells versus spore genetic material also?
It's a really interesting question.
Mm-hmm. So
go ahead, Yuri.
I can talk to the detection really quick. So this detects the presence of genetic material, so DNA. So no, it cannot differentiate between dead cells and live cells, right? If there's DNA from dead cells floating around, it does detect that. Generally, though, the presence of dead microbes indicate that there are microbes growing somewhere. And then same for spores and stuff like that, right? If there's genetic material in there, we're going to be able to detect it. I hope that answers the question.
Thank you. We have a question from, see if I'm pronouncing this right, Mosen Akor. Is there data to show the accuracy of the method compared to qPCR samples, for example?
Yeah, so we're using a method that is fairly similar to qPCR testing, except it's more sensitive. So yes, there is data to show comparison between our testing method and qPCR that shows that our testing method can be more sensitive than qPCR. As far as accuracy goes, it really depends on how you develop these tests, but we found that we basically see little to no false positives or false negatives with our tests, similar to what you would see with qPCR. So they're very, very similar in a lot of ways.
Great.
And we can handle dirtier samples too. You don't have to do a DNA extraction like you would for a PCR.
Diane has her hand raised. I'm going to go ahead and unmute Diane. Diane, you should be able to.
I just want to make that the question which I asked earlier a little clearer. At this point, the presence of a microbe, sets of microbes, or a consortia in the gas does not indicate corrosion on the pipe wall. These are two different things. I mean, this is the biggest issue that we have. I love rapid testing. It's used in medical. In fact, they're fancier than this in medical environments. But I want to make sure that that's the piece which is driven home. It isn't a number. It's the coalition of factors which create an environment that cause microbes, which are kind of secondary to the whole issue, to bloom and to cause corrosion. And this is a very dynamic situation. It isn't static, can change within no time at all. I just want to help the industry frame this a little better, rather than saying you have this equals this. That's what I want to ask. So what's your intent?
Yeah, no, that's a great point, and you're right. Like it's not as simple as microbes are there, so you're going to see corrosion, right? And that's why we say we are working on a device that can detect the presence of microbes, but we're not detecting corrosion, right? So there's that distinction there, and you're right. It's important to kind of make it clear that we are testing for the presence of these microbes, that you can then generate data to infer other stuff from it, right? But we're not testing for the presence of corrosion. You're completely correct there.
Which is why we work with the corrosion the folks who are doing the integrity testing and making those decisions on what biocide should be used and if it's appropriate. We're a tool within that. We're giving them more information faster so that they can make those decisions in an intelligent manner.
Thank you for your question, Diane, and for your clarification. We have another question from Vincent Arias. And just a reminder to all the attendees, you still have time to raise your hand if you want to speak directly. Vincent asked, is there a detection difference if you sample and test immediately versus sample and test the next day?
So yes, generally, if you kind of just leave your sample sitting around either outside at room temperature or in the freezer, stuff starts degrading, right? DNA is fairly stable, so for the most part, you're gonna find most of the same stuff, but there might be some loss of signal. Yeah, basically, the sooner you can test, the better, because as soon as you draw the sample out of your system, stuff starts degrading, right? So you're gonna lose signal basically the longer you have it sitting around.
Thank you. And we do have a few more questions, but I'm just going to take one more, and any additional questions, we will display a slide at the end where you can email people directly for the questions, or you can email rddinfo@SoCalGas.com. Let me go ahead and ask this final question from Mohsen Akor. Is the kit for rental or for purchased? And what is the approximate cost, rental or purchase?
Yeah, we're moving towards commercialization. Yeah, we are going to be rolling out here in Q1. So go ahead and reach out to me at mteagle@biovind.com, and we'll talk about how to do a demo with you. Ideally, we'd want to go out to your site and figure out what's in your system first. We'll do an NGS test, and then we develop a custom cartridge for you. And then those are going to be about the same cost as a culture test, as those bug bottles would be.
Perfect. Thank you all so much, Mark and Yuri. That was great. Tyler, you can go ahead and display the closeout slide. If anybody has any additional questions, more information can be found there. And final thank you to all the attendees. Have a great day.
Thanks, everybody.
Thank you, folks.